in order to handle the cell cultures, you will need to adhere to the good laboratory practice in the lab. So for example, the personnel will need to wear protective clothing, need to wear gloves, goggles, okay, and practice the set of aseptic skills which is required to culture the cells. So why do we need to practice this aseptic technique? This is to reduce the risk of exposing the worker to potentially infectious agents in the cell culture, as well as to prevent contamination of the cell cultures. So the nature of the work must conform to the regulatory guidelines and legislation. So aseptic technique and the proper use of laboratory equipment are very essential when working with cell cultures. So you need to ensure that the reagents and mediums you are using are sterile. Okay, there's no contamination. You always need to swab the work surface with 70% ethanol before and after you have finished your work. So what happens when your cell cultures are contaminated with microorganisms like bacteria, virus? So this can inhibit the cell growth, or it can kill the cells, and most often it will lead to inconsistent results and performance. So this is the diagram where when you observe the cells under the microscope, under high magnification, you will see there's a lot of rod-shaped bacterial cells here observed in the diagram B, where it is contaminated with E. coli. So what are the potential sources of contamination? So this might be due to the poor handling or the techniques used to culture the cells. It may be caused by contaminated mediums or reagents or the equipment that you are using. So there also might be microorganisms present in the common storage areas like incubators, refrigerators, or the laminar flow hoods, as well as on the surface area of the skin of the worker or in the cultures coming from other laboratories. So the workers will need to ensure that all sources of contamination are identified in cases where their cultures are contaminated. So what are the reagents or culture medium that we can use to culture the cells? Though this will depend on the cell line that you are culturing. Each cell line will have their own growth requirements, so they will need specific growth factors, cytokines, hormones that may be used for culturing the specific cell lines. So these are the physiological conditions that we provide to the cells. So these include common basic medium like minimum essential medium, MEM, Dulbeckel's modified Eagle medium, DMEM, RPMI, and the HAM-F10. So all these medium contain a mixture of various nutrients, for example, amino acids, glucose, vitamins, and others, and they are available in the form of powder as well as liquid from various commercial suppliers. So we will culture the cells in the CO2 incubator. So this incubator is different from the ones that you use to culture bacteria. So this is a incubator with water jacketed, which means that they will have the water tray beneath here where it is filled with the deionized water and it is supplied with the 5% of the CO2. So we'll culture the cells in disposable dishes or flasks. So these culture dishes or petri dishes is different from the one that are used for bacterial culture. So these cell surfaces of the plates and the flasks are being treated especially for the culture and attachment of animal cells. So cells in this case, they are treated by electric ion discharge, plastics with a net positive charge. So how do cells actually attach to the bottom of the plastic dishes or the culture flasks? So cells will normally release certain type of matrix products. So these matrix products will adhere to the substrates and provide ligands for interactions of the matrix receptors such as integrins. So these ligands will attach to the surface of the plastic dishes. So you will treat the substrate with matrix products such as collagen, fibronectin to promote the adhesion of the cells. So how do we maintain the cell cultures? So cell cultures, we will need to always change the medium or subculture the cells when they have reached confluency, which means that once they have occupied all the available space of the culture dishes or the flasks, we will need to divide them and separate them into different culture dishes. So these cultures should be examined regularly. So you need to observe under the microscope to check for any signs of contamination and to determine whether you need to change the medium or you need to subculture the cells. So this is the growth profile of the cells. For example, they will go through the lag phase. Slowly they will increase in number to the log phase and before they reach the stationary phase. So the arrows here indicate the procedures that you will be doing along the way. So this is once after the cells have been subcultured. Okay, so when they move into the log phase, you might want to change the medium where the cells are actively metabolizing. Right before the cells enter the stationary phase, you will need to subculture the cells before they reach the stationary phase. So how do you subculture the cells? So for those adherent cells where the cells adhere to the bottom of the culture dish, you will need an additional process known as trypsinization using the trypsin enzyme. For suspension cells, it is relatively easier because you will just need to divide the cells into half and replace with the medium. So for trypsinization is only applicable to adherent cell types. Okay, so this is before and after. So you can see from here that the cells are very packed, which means that they have already occupied all the available space here on the left. So once they have reached this condition, you will need to subculture the cells, which means you need to divide them into separate culture dishes to expand the cells, or you want to throw away the cells if you do not want to use them immediately. So in this trypsinization process, you will incubate the cells with the trypsin enzyme. So this trypsin enzyme will digest the substrates between the cells and the cell at the surface of the flask so that they are broken. These linkages are broken where you release the cells to become a single cell, okay, before you transfer them into a new culture dish from here. So the adherent cells will need to detach before you transfer them into a new dish or a new flask. So what is cell banking? So you will need to stop or cryopreserve the cells from time to time to maintain the cell stock. So this is especially important where those cell lines are very small in numbers or you want to use them in the future. So you do not want to use the cells immediately now. So maybe for the next 3 months you will not be using it. You will want to stock it to frozen it or cryopreserve it. Maybe you will need to use in a few months later. So the process of storing these cells is known as cryopreservation. So there are two types of cell banks where they call it as a working cell bank or the master cell bank. So you normally only take out cells from this working cell bank, while the master cell bank is a repository where you store the cells in this master cell bank, and you would not be taking out of the master cell bank unless it's very necessary. So the cells will need to characterize. Okay, for example, you have a new cell line, you will need to identify what type of cell line is it. It belongs to the new subtype of the cancer cells. You will need to identify whether are there any cross-contamination between different cell types. So you need to confirm the cell line identity. So you will need to characterize the cells. So what are the techniques for characterization? So you can look into the chromosomal analysis to look at the types of enzymes that the cells are expressing, the types of proteins they are expressing, serotyping, or to sort the cells based on the surface markers.